Okay, folks, let's get started. For the last time, we're going out on a very nice day. So, a couple of things. First of all, I'll say it again probably at the end, but I've really enjoyed working with you guys and getting to know you. This has been a really neat class. You have been the most unusual class I've ever had. I've said that, and it's true. Not just because you giggle when I say things, but because usually you don't react at all, and that's really odd. But I'm happy. I'm very happy with you, and I've gotten to know several of you, and I really appreciate that. So, very happy to have that happen. I apologize. I am not going to be able to do a review session for you. I'm tied up tonight. I'm tied up tomorrow, and I'm hopping on a plane tomorrow night, and there's no way I can do it. What I will do, however, is I will make links for 2 or 3 other review sessions that I've videotaped before and link those for you on the page, okay? So that's one. Number 2, if you have questions that aren't answered on the review sessions or whatever, please feel free to give me a holler, okay? And I'll even give you my cell phone number, okay? So 541-908-4465. If you have questions, give me a holler, okay? Everybody with me? All right, so I apologize, I just can't do that. My wife, Indra, which I think some of you know, will be here giving the exam to you on Tuesday. Is it noon? Is that right? Okay, I think that's right. In any event, check the schedule. The schedule has the schedule page has it correct. That will be there. If you didn't get a note card, you need to get one today because I'm leaving town, and you have to bring a note card with your name on it that you got from me, even if you're not going to use it. So make sure before you leave today that you get a note card because this is the last opportunity. Okay? If you don't have a note card like that, you will lose points. Yes? Front and back. If you can write on the edge of it, you are welcome to try to do that as well. But everything on the note card has to be handwritten. Okay? I want to emphasize that. Everything on the note card has to be handwritten. Okay? No printed note cards. Okay? What's that? The final is right here. Right here. Other questions? Okay, well, if we get through the material, I've got a couple of fun songs for you that I hope you'll enjoy at the end. And we've got some ground to cover. So let's go through and cover that ground. I mentioned to you that uric acid was a problem because uric acid can form crystals, and it commonly forms crystals in the lower part of your anatomy, most commonly in your big toe, but it can also be in other areas like ankle and so forth, and cause severe pain as a consequence of that. Uric acid is a breakdown product of purine metabolism. So purines include adenosine, guanosine, and when those break down, they can break down and form uric acid. If the scheme is working okay, you break those molecules down and then into uric acid, and then uric acid itself breaks down ultimately to form urea inside of you. Okay? As I noted the other day, dalmations and birds actually excrete uric acid. We don't do that. But you can see that there's a fair amount of nitrogen there. There's also a fair amount of nitrogen in urea. And so these are effective ways of getting rid of excess nitrogen in the body because, excuse me, as I noted last time, too much nitrogen is toxic. Okay. That brings me to something important called the urea cycle. So the urea cycle is another cycle that's in our body, and no, we're not going to pay as much attention to this cycle as we did the citric acid cycle. But the urea cycle is something that occurs between the cytoplasm and the mitochondrion. Some of the reactions occur in the cytoplasm, some of the reactions occur in the mitochondrion. Okay? Let's see. What do I want to say? It has several interesting features. First of all, you can see the names of the molecules there. This is the mitochondrion up above. And you can see 2 of the reactions occurring inside of the or actually one of the reactions occurring inside of the mitochondrion. The formation of urea comes from the breakdown of the amino acid arginine. Okay? So this is actually arginine right here. And when arginine breaks down, it leads to production of urea. And then another compound that's labeled here called ornithine. Now, why do I care about that? The only reason I even mention ornithine is that it's an example of two amino acids that we have in our body that are alpha amino acids, just like the amino acids in proteins, except for neither one of them appears in proteins. Ornithine is the first. The second is the compound it's converted into, which is citrulline. Okay? Citrulline is used sometimes in energy drinks because there are people who have this idea that citrulline provides a lot of energy. Yes, ma'am? Sure. So we're back. That was a sound effect. I was rewinding there. You thought I totally lost it, right? It's the last day of class, guys. I mean, I've got to make you laugh somehow. Nobody ever reacts to my stuff. Yes. The urea cycle. You want it slower? All right. The urea cycle is a cycle that occurs, okay, in our cells, and the function of the urea cycle is to help you balance and get rid of excess nitrogen. Okay? Help you to balance and get rid of excess nitrogen. It involves several intermediates, and the reactions occur partly in the cytoplasm, which is down here in the bottom, and partly in the mitochondrion. Okay? We're not going to memorize all of the molecules. We're not going to memorize this goes to this, goes to this. We're not going to memorize the enzymes. But I am wanting to make you aware of a few of the molecules that are in there. And that's what I was trying to do in my moving at the speed of light lecture there. Okay? All right. So, some of the molecules that are of interest, the amino acid arginine. Arginine, okay, is the precursor of urea. That is, you can clip off the last carbon, this carbon right here on the end, and when you do that, you create urea. Okay? By removing that carbon and forming urea, what's left behind is another compound called citrulline. I'm sorry, called ornithine, sorry. Ornithine, right here. Okay? And ornithine is one of two amino acids in this cycle that are not found in proteins. Two amino acids in the cycle that are not found in proteins. The other one is citrulline. You can see it over here. Okay. Now, like other cycles, like the citric acid cycle that you've already seen, things that go in a circle mean things can come in at various places, things can leave at various places. And so if the cell were needing, for example, to make arginine, it could suck off the arginine off of this cycle and use that. If it had excess arginine, which would indicate that the body had excess nitrogen, then it could feed that into the cycle, and urea would be produced, and then that would be excreted. Okay? So the cycle performs that balance function that's very important for this process, or for our bodies. Does that help? Okay. All right. I want to turn our attention to the synthesis of purine nucleotides. And I'm going to go through this very briefly. The metabolism of nucleotides is a fascinating topic. Okay? It's an absolutely fascinating topic because the cell and the body goes to a tremendous amount of effort to make the proper amount of nucleotides. If the proper amount of nucleotides is not made, let's say they make too much dCTP and not enough dATP. Okay? If that happens, that cell will be much more likely to have mutations because the proper amount of nucleotides is necessary for accurate DNA replication. Now, I'm not going to talk about all of the different mechanisms that are involved in controlling that, but suffice it to say there are many involved in controlling the levels of nucleotides inside of cells. Well, this slide isn't to show you about the levels of nucleotides inside of cells, but rather to show you the source of the various atoms of the purine bases. So this is a structure of the purine bases, and you can see where the atoms ultimately come from. This carbon on top, for example, comes from carbon dioxide. The nitrogen comes from aspartic acid. The carbon comes from one of those folates that we talked about the other day. There's a nitrogen from the amino acid glutamine. There's a central part that comes from glycine. Okay? Now, you're not going to memorize this. That's not my purpose in showing you that. My purpose in showing you this is to remind you of something that you always hear when you read news reports about meteorites falling to Earth, and they have things in them. Okay? It's possible to make, in just ordinary chemical reactions, nucleotides from very, very simple compounds. Okay? Life on Earth evolved because this simple chemistry allowed for things like DNA, RNA, and proteins to form from what was on the primordial Earth. Okay? Very, very cool setup. I'll show you a similar one for pyrimidines, and pyrimidines are even simpler than are the purines. Okay. Well, there's a lot of steps in making nucleotides. And you guys have seen, in my opinion, enough steps this term. Unless there's somebody who would like to see a few more steps. A few dance steps? No. No, I didn't hit happy hour before I came to class. Okay. I wish I had. Maybe you wish I had too, I don't know. All right. But the point of this is to tell you a couple of things. First, there's about 10 steps that it takes to start from scratch and make purines. It takes about 10 steps to start from scratch and make pyrimidines. Okay? The first thing that we make in the purines that looks at all like a nucleotide, okay, is this thing called IMP. It's a nucleotide called inosine. Does anybody recall having seen inosine earlier in the term? There was a molecule that I showed earlier in the term that had, instead of the nucleotide G, C, etc., it had a nucleotide I in it. And I very briefly mentioned it. Does anybody recall that? For 2 points of extra credit? No? Okay. The place where you saw it was in the anticodon of the tRNA. And that inosine, that I, could base pair with multiple things in the codon. I very briefly mentioned it. I didn't have a chance to say much about it in class. In any event, inosine is a purine nucleotide. However, it does not make it into DNA. Okay. Why do I show it to you? Well, I show it to you because inosine is the branch point between the synthesis of AMP, which is down here on the left, okay, and GMP, which is over here on the right. These are both purine nucleotides. Okay? Now, these nucleotides are interesting, and I'm going to tell you a little bit about regulation at this point so you can understand some of the balances that are in place in cells to make sure that cells don't get too much of one or the other. Okay? How does this work? Well, let's go down the left side. Let's look at the synthesis of AMP, starting with IMP. Okay? AMP is made, okay, using energy from GTP. Now, if you think about it, that sort of makes sense. Because if you didn't have any ATP, and you needed ATP to make AMP, you'd kind of be hosed, right? So fortunately, there's enough GTP to allow for the synthesis of AMP, which can ultimately be made into ATP. Second, okay, AMP will feedback inhibit this enzyme to stop it from making more AMP. So if the AMP concentrations get too high, then the synthesis of AMP will be stopped. And no, I'm not asking you to know the names of the enzymes. But you should know that AMP feedback inhibits its own synthesis. Let's look at the other side. On the other side, the synthesis of GMP requires energy from ATP. And further, GMP inhibits its own synthesis. Okay? Now, there is one enzyme whose name you should know that's important in regulation. And that enzyme has a long name, unfortunately. It's the very first enzyme in the overall pathway. And yes, I think you should know the name. It's called PRPP, amido, A-M-I-D-O, transferase. PRPP, amido transferase. Now, why do I tell you that enzyme? That catalyzes the first reaction on the pathway that leads to purines. And I tell it to you because its regulation is really interesting. That enzyme is feedback inhibited by AMP and GMP. And notice I said and. It takes both of those to bind to PRPP amidotransferase and turn it off. If only one is abundant, let's say only AMP is abundant, the enzyme will only be partly inhibited. It's the first time you've seen that example happening. Only if both are present will the enzyme be shut off. Now that turns out to be important because if you think about it, let's say you had a lot of AMP, but you had virtually no GMP, you would still want to be able to make GMP, right? So starting that process would be important, and the feedback inhibition for AMP would shut off this side, but it would not shut off this side, and you would continue to make GMP. Okay, so you're starting to see some of the balances that cells are going through in order to make sure they have the right amounts of nucleotides. Questions on that? Get out of here, right? Okay. All right. That was an evil laugh. Oh, you don't want to see that. All right, we'll turn that off. This is part of what you've already seen before, all right? These are the purine nucleotides, and they're breaking down ultimately to make uric acid, all right? We'll leave it at that. Okay? Another thing that's important in the synthesis of nucleotides is something we call salvage synthesis. What's a salvage yard do? I'm going to start talking backwards if you guys don't answer. Okay, so they're taking parts of automobiles, they're taking automobiles that are of no use and salvaging the parts out of them, right? So cells do that with nucleotides as well. They can salvage, let's say, a base that has lost its sugar, and they can use that base to make a full-blown nucleotide. That's called salvage synthesis. Okay? It's called salvage synthesis. Salvage synthesis would seem to be fairly trivial, but it turns out to be very, very important because if cells are lacking the enzymes necessary to do salvage synthesis, people who have deficiencies in those enzymes end up with severe neurological problems. The one I always give as an example, people kind of go, ooh, all right? One of the examples for lack of salvage synthesis is a genetic disease called Lesch-Nyhan syndrome, and I'll show the thing on the screen for you. All right? There's the spelling right there, Lesch-Nyhan syndrome. Lesch-Nyhan syndrome, okay, results in a deficiency of one of the enzymes necessary for salvage. Okay? People who have this disease literally eat everything like their fingers. They will chew their lips off. Okay? They have bizarre behaviors that arise as a result of that. And it happens because they're deficient in one of these enzymes for salvaging nucleotides. They just have a craving to do it. There's a neurological problem, and obviously some wires crossed in the brain. But in severe cases, they have to be strapped down because they will literally eat their fingers off. And when they strap them down, they're eating their, yeah. What's that? Not having Lesch-Nyhan syndrome myself, it's hard for me to say. So, okay, we're doing well here. Pyrimidines. We've just covered purines, and now we're moving into pyrimidines. Life is good. Okay? As I said, pyrimidines also take about 10 steps to start from scratch to make the first pyrimidine. The first pyrimidine that's made, okay, inside of cells is UMP. UMP. That's the first one. And if we look at the things that it goes into making pyrimidines, again, we see some very simple compounds. There's bicarbonate. There's the amino acid glutamine. Okay? There's aspartic acid. Right? There's coenzyme Q, which you've seen from other things, donating electrons as necessary. But very simple things that go together to make pyrimidine nucleotides. There's a very important regulatory enzyme for pyrimidine synthesis, and you know what it is. What is it? Slurpee? Did I hear Slurpee? I'm not going to get very far with this line of questioning, am I? It was an enzyme that was inhibited by CTP, it was activated by ATP, and it was activated by aspartate. Do you recall that? Say what? No, not a dehydrogenase. Anybody remember ATCase? Is that what you said? If you said a lot, you would have gotten 2 points, but too bad. ATCase. ATCase is right there. It catalyzes one of the very first steps in the process. And the end product of this pathway is ultimately CTP. So CTP comes back, feedback inhibits ATCase. Notice here that a pyrimidine turns it off, and a purine turns it on. And that makes very good sense, because if we have too many purines and not enough pyrimidines, we want to start the synthesis of pyrimidines. And if we have too many pyrimidines, we want to turn off the synthesis. ATCase is another enzyme that helps in the balance of the relative amounts of nucleotides that's being made. Okay? All right. Well, I told you that the first nucleotide that was made was UMP. And when we looked at the purines, you saw we made AMP and we made GMP. Now we've got UMP. How do we get the triphosphates? Okay? Well, there's a couple ways we can get them, but the way that we're going to talk about here are a couple of kinases. Kinases, I hope you remember, are enzymes that put phosphates onto things. They use the energy of ATP to put phosphates onto things. Okay? UMP is converted into UDP by something we will call mononucleotide kinase, mononucleotide kinase. Right? Mono meaning that starting material is a monophosphate. Kinase puts a phosphate onto a monokinase, onto a molecule with one phosphate. We end up with a molecule with two phosphates. We will just call this a actually let's give it a mononucleoside kinase, a mononucleoside kinase. Okay. What's that? Well, you can call it either thing. A mononucleotide is the same as a mononucleoside phosphate kinase, phosphokinase, right? So, all right. How about? Should we give it a name? No. Okay. All right. It's a kinase, and it's a kinase that works on nucleotides with one phosphate. It turns out that there's a specific one for each of the nucleotides, and we're not going to give each one names. All right? But we are going to know the name for the one that puts the phosphates onto the nucleotides that have two phosphates. This one's very easy. Okay? Nucleoside Diphosphokinase, which we will call NDPK. NDPK. NDPK works on every nucleoside diphosphate. So if this is UDP, NDPK will work on it. If this is GDP, NDPK will work on it. If this is ADP, NDPK will work on it. If it's DGDP, NDPK will work on it. So one enzyme works on all of the diphosphates, whether they're deoxy or ribonucleotides. Notice in each case, the energy for this comes from ATP. Okay? And we don't need to mess with that. And I know you want to mess with that, but we're not going to mess with that. And the catabolism, the breakdown of pyrimidines, is a lot less interesting than the breakdown of purines. Pyrimidines don't break down into things like uric acid that cause problems for us. And there are some metabolic syndromes associated with some of the deficiencies in the breakdown of those, but none of them are going to be of interest to us. Everybody's starting to smile. Okay? Here's some words you've been wanting me to say for a while, and that is the last thing we're going to talk about in this course. It's true. Is how we make the deoxyribonucleotides. That is the molecules that go into making DNA. How do we make those? It turns out that the deoxyribonucleotides, at least 3 of them, are made directly from ribonucleotide diphosphates. Okay? So, for example, the precursor of DGDP, that would be deoxyguanosine diphosphate, the precursor of DGDP is GDP. The precursor of DADP is ADP. The precursor of DCDP is CDP. You start to see a pattern? Where does T come in? Okay. T is a little bit more complicated. I'll talk about it in a minute. But it ultimately comes from UDP. Okay? Now, before I tell you how T is made, I want to tell you about the enzyme that makes the ones I've just told you about so far. The enzyme that converts ribonucleoside diphosphates into Deoxyribonucleoside diphosphates is an enzyme called Ribonucleotide Reductase, which you are welcome to call Rnr. So the enzyme that's converting GDP to DGDP is Rnr. The enzyme that converts CDP to DCDP is Rnr. The enzyme that's converting ADP into DADP is Rnr. Everybody with me? If I have DADP, how do I get DATP? What enzyme? No, not Rnr. Rnr is going to convert from ADP to DADP. If I want to go from DADP to DATP, what do I need? NDPK, right? Say it again. That's fine. You guys should have been more vocal earlier in the term. Everybody would have come out of this thing with 100%, right? All right. So let's back up. All right? So let's first of all think about the topic that we're talking about is the synthesis of the deoxyribonucleotides, right? So far, I've shown you how to make the ribonucleotides. That was how we had the purine pathways and the pyrimidine pathways, right? The products of those pathways are ribonucleotides. CTP is a ribonucleotide, okay? ATP is a ribonucleotide. GTP is a ribonucleotide. UTP is a ribonucleotide. So everything we've talked about before I got up to Rnr, everything up to that point was talking about ribonucleotides. All right? So now the question is, how do we get the deoxyribonucleotides necessary to make DNA? So let's do it backwards from what I said before. Let's start with the enzyme. The enzyme is called Ribonucleotide Reductase, Rnr. Okay? That enzyme is essential for making deoxyribonucleotides, Rnr. Without that enzyme, you can't make deoxyribonucleotides, you can't make DNA. It's that simple. Okay? The question then is, how does Rnr convert a ribonucleotide into a deoxyribonucleotide? The way it does it is by starting with diphosphates. Diphosphate. Starts with diphosphates. This includes ADP, GDP. Notice we're starting with ribose, CDP, and UDP. Those are our starting materials. Each of those can be converted by Rnr into the respective deoxy forms of those. Okay? Into the respective deoxy forms of those. You can see that happening right here, right? UDP, CDP, GDP, ADP. We lump those together and we say they're known as NDP, where N is any of those bases, right? NDP, when catalyzed with ribonucleotide reductase, converts NDP into DNDP. That means A goes to DA, G goes to DG, C goes to DC, and U goes to DU. Everybody with me? So that's how we're making the deoxy forms. Then, once we've got the deoxy forms in the diphosphate, it's simple to convert them to the triphosphate because now we use this called nucleotide kinase. It's another name for the same enzyme, NDPK. NDPK. Okay? Now, the only thing we're lacking is understanding how this enzyme controls things and how thymidines are made. Does everybody understand this? Other questions on this? Yes, ma'am. This is NDPK. Yes. Because NDPK will work on any diphosphate and turn it into any triphosphate. Okay. Well, what's the difference between them? Well, you can see the chemical difference down here. There's ribonucleoside diphosphate. Base, ribose, diphosphate. Here's the equivalent deoxy form. Base, deoxyribose, notice there's an H there instead of an OH there, and diphosphate. So what the enzyme is doing is it's essentially removing this oxygen and replacing it with a hydrogen. Okay? That's what Rnr catalyzes. Yes, Miriam? Okay. I read her mind. Okay. All right. Well, at the risk of totally confusing things, and whenever you say that, you know you're in trouble, right? At the risk of totally confusing things, I want to say just a very brief word about how Rnr is regulated. We've been talking about balance, and we've balanced purines, we've balanced pyrimidines, and now we've got to balance the deoxyribonucleotides as well. Rnr performs that function. Now, the regulation of Rnr is very complicated. And I'm going to give it to you in very simple terms. Basically, high levels of purines favor production of pyrimidines, and high levels of pyrimidines favor high levels of purines. That is, production of high levels of purines. So if you have too much of one, the other one is favored in terms of being made. Now, I'm not going to expect you to know the things that bind to the different sites, but there are two sites on the enzyme that are important to know. One's called the activity site. As you might imagine, an activity site determines if the enzyme is active or not active. And this one's very simple. Okay? If DATP binds to the activity site, the enzyme gets turned off. Think about that. DATP is a deoxyribonucleotide. Too much of it shuts the enzyme off. If ATP binds to the same site, it turns the enzyme on. ATP is an indicator of energy. Cell has plenty of energy. It wants to divide. It's going to need nucleotides. Everybody got that? The last site is called the specificity site. These are different places on the enzyme. The specificity site is where the enzyme decides if there's too many purines or too many pyrimidines. The specificity site. Okay? Almost there. Any other questions on that? We got one very last topic. Okay. The last topic is relevant because it has medical relevance for us. And that's the synthesis of the thymidine nucleotides. Okay? Don't worry about the structures. But it's interesting how they're made. Okay? The starting material for the synthesis of thymine nucleotides is UDP. Because, again, Rnr is going to work on UDP. And when Rnr makes UDP, it converts it into DUDP. Just like it converts GDP into DGDP or CDP into DCDP, it converts UDP into DUDP. Okay? Now this figure actually cheats because it says something, it's missing a step, but I'm not going to include the missing step. DUDP gets converted into DUMP, which I really like because it says dump, which at this point is about what you're ready to do in the class. It's just dump, right? Okay. There's an enzyme called thymidylate synthetase, which, yes, you should know, that converts DUMP to DTMP. This is the most important enzyme in this process. Converts DUMP to DTMP, thymidylate synthetase. Notice that it's using a folate to do that. I mentioned this reaction on Wednesday. The folate is donating a methyl group for this reaction to occur. Okay? The folate is donating a methyl group that converts DUMP into DTMP. Notice the methyl group right here, the methyl group right here, and the no methyl group on the DUMP. So this methyl group is coming from the folate. Folate turns out to be a target for anti-cancer drugs. Okay? This is a complicated figure. We're not going to mess with the figure, all right? But I'll show you something on the figure that is interesting relative to cancer, all right? Folates have cycles of their own in our cells, meaning that they get made, broken down, recycled, et cetera, and the thymidylate synthetase is using a part of that cycle up here. It's converting DUMP to DTMP, and it's starting with that, and it's producing that. All right? You remember the story with NAD and NADH? We've talked about it several times now. Cell has a limited amount of that. If all of the NAD is converted to NADH, there are consequences for the cell, right? Similarly, there's this limited amount of folates present in cells. If this guy here gets converted to this guy over here, and the rest of the cycle doesn't happen, then what's going to happen is there's not going to be anything to make more nucleotides. And if you can't make more nucleotides, you can't make DNA. And if you're a cancer cell dividing more rapidly than a regular cell, if you can shut down their supply of nucleotides for a short period of time, you're going to be more likely to be able to kill that cancer cell. Why do I show that here? Well, it turns out that there's a drug that will inhibit this enzyme here called DHFR, dihydrofolate reductase. The drug is called methotrexate. You may know somebody who's taken it for chemotherapy. Metho, M-E-T-H-O-T-R-E-X-A-T-E. By inhibiting this enzyme, the cycle doesn't get completed. Consequently, the cells can't make folates. Consequently, they can't make thymidine. Consequently, they can't make DNA, and they can't divide. You give this drug to a person for a short period of time, because if you gave it to them for a long period of time, you would kill them. But the idea is that by giving it for a short period of time to a cancer cell that is more susceptible to changes in nucleotides, the cancer cell will die faster than a regular cell will. Okay? It's a fairly harsh treatment for cancer in its appropriate doses. But for a rapidly dividing cancer, it can have some fairly welcome effects in terms of knocking down the cell synthesis. Okay. Are you guys ready for a couple of songs? All right. So I have two songs for you today. The first one is a song I've never sung to my class. It was the one I had hoped I would have a recording of for you, but I don't have it, so we're going to sing it. It's called At Dairy Queen, and it's for the nutrition majors in the class. And the second song is one, by the way, the Dairy Queen song is not a very lively one, so it's kind of like, okay. So I decided we had to finish with a lively one, and so I have a very lively one to finish with after that. So we got 2 songs. First song. Oh, that's the wrong one. All right. So this is to the tune of an old song way, way back in the 70s called At 17. I'll start it, and then I want you to please join me. You'll see the melody is fairly simple, okay? Will you do that? I'll put an extra credit on the exam, if you will. Make it 2. I saw the light at Dairy Queen while eating chocolate ice cream. That those who crave their sweeties, lad, develop diabetes bad. The sugared things they sell to you seem innocent, but it's not true. They'll raise your insulin and thus cause problems for your pancreas. We buy the stuff and never think of how we're almost on the brink of getting some publicity for sugar-based toxicity. That's all because the fast food scene is mostly sugar, salt cuisine. We're bursting at the seams at Dairy Queen. At Mickey D's I order fries that come to me all supersized. I eat them up until I'm full, since ketchup is a vegetable. At Taco Bell I must disclose, tortilla shells have GMOs, and cooking oil unnaturally is poisoned so trans-fattily. Remember this when picking food, processing can leave you screwed. In colas you will get a dose of syrups laced with high fructose, and diet ones are all replete with chemicals like NutraSweet. I wonder what it means at Dairy Queen. It's no surprise to folks like me that problems with obesity are rooted in the food supplies, combined with lack of exercise. Of course, it makes a lot of sense to read each package's contents, then use the info that it gives for cutting out preservatives. If we kid ourselves, we'll pay the price with poor nutritional advice, imagining ourselves okay, forgetting how much that we weigh. There's no ignoring nature's call when it comes to cholesterol. No ice cream cone is free at Dairy Queen. Oh, there we go. Okay. All right. To who? There we go. I will sell it to anybody who wants to buy it. Would you like to buy it? Okay. Now, the last one is probably the most popular song that I have to sing at the end of the term, and it requires a lot of help from you. It's to the tune of an old John Denver song called Thank God I'm a Country Boy. And it's called Thank God There's a Video. And it really helps if you clap. And you got to sing along with me. Ready? There's a bundle of things a student ought to know, and Ahern's talk isn't really very slow. Learning ain't easy, the lectures kind of blow. Thank God there's a video. We'll be gone through the cycles and their enzymes too, studying the regulation, everything is new. Gotta admit that I haven't got a clue. What am I gonna do? So I got me a note card, bought me a strier, got the enzymes down and the names he requires. Hope I can muster up a little more desire. Thank God there's a video. Just got up to speed about the NAD. Protons moving through complex V. Electrons dance in the cytochrome C. Gotta hear the MP3. Fatty acid oxidation makes acetyl-CoA inside the inner matrix of the mitochondria. Very complicated, I guess I gotta say. Thank God there's a video. So got me a note card, bought me a strier, got the enzymes down and the names he requires. Hope I can muster up a little more desire. Thank God there's a video. Replication's kind of easy in a simple kind of way. Copying the bases in the plasmid DNAs. G's goes with C's and T's go with A's, thanks to polymerase. And the DNA is a template for the RNA. Helices unwinding at TA. Termination happens and the enzyme goes away. Don't forget the poly A. So got me a note card, bought me a strider, got the enzymes down and the names he requires. Think I can muster up a little more desire. Woo! Thank God there's a video. All right, thank you guys. Good luck to you. Stay in touch after you leave, okay? Woo!